My name is Klapperich, and I'm a professor in manufacturing engineering and biomedical engineering at Boston University. And I'm the director of the Biomedical Microdevices and Microenvironments Laboratory. We work primarily on making disposable diagnostics for applications in resource-limited settings. So we use microfluidics in order to make those devices. We're interested in actually two fields of study. Primarily, we're interested in looking at how biomolecules and cells interact with plastics, polymer surfaces. That's our major umbrella. That work is embodied in microfluidic applications for disposable diagnostics. So in the laboratory, what we make are microfluidic devices in thermoplastic materials. We don't typically use PDMS, which is the rubbery material that most people use to make microfluidics in the laboratory. We make our devices out of thermoplastics. We're interested in using those thermoplastic devices to do molecular biology at the microscale in such a way that that molecular biology could be done in the field far away from a centralized laboratory. How that works out in the lab is we're making little plastic cards, and inside of those little plastic cards are solid phase extraction columns or cell lysis columns or mixing columns. We also do polymerase chain reaction on the chip as a detection technique. All of these things are integrated into a small device that eventually, in the long-term outlook of the research, would end up being an integrated device that would be able to be handheld and carried out into the field or into settings where a large-scale laboratory with all the attending centrifuges and heating